Recall what prion are: misfolded forms of a normal protein, PrP-SC versus PrP-C, that act as a template, inducing other normal proteins to adopt the same abnormal conformation. This process does not involve nucleic acids and is essentially a self-propagating misfolding. Next, recall what molecular chaperones do: they assist correct folding and prevent aggregation. Chaperones, e.g., HSP70, HSP60, bind exposed hydrophobic patches on partially folded or misfolded proteins, shielding them from aggregation. They use ATP hydrolysis cycles to promote correct folding or refolding. Now let's evaluate the options. Option A describes prion as templating misfolding without nucleic acid, and chaperones as preventing misfolding by binding hydrophobic regions and using ATP. This matches standard definitions. Option B is incorrect because prion do not use ATP hydrolysis. Option C reverses roles: chaperones do not cause misfolding, prion do not restore correct conformation. Option D is incorrect: prion are proteins, not RNA. Option E is too absolute and partly wrong: many chaperones also act on misfolded mature proteins. Thus, the correct answer is A.